Hello, everyone, and a very good evening to one and all. I'm very glad that I have this opportunity of bringing people to come and speak up on behalf of the 3D bioprinting community. So, thanks a lot to everyone who's joining and whoever is yet to join. Please, share links across, talk to people, share whatever required links, datas required on this presentation. So, uh, our session has been on the realistic approach of 3D bioprinting, and we are on the day three of our session. And our day three is mainly for our guest speaker, and today's topic is going to be 3D bioprinting in cardiac tissue. And before I start, I would like to introduce our guest speaker, who's Shreya Mehrotra. And talking about Dr. Shreya Mehrotra, she's a postdoctoral fellow at IIT Kanpur. She has pursued a PhD in the field of tissue engineering and regenerative medicine from IIT Guwahati. And she's extensively worked in areas like biomaterial fabrication, 3D and 4D bioprinting, microfluidics, use of cardiac tissue printing in regenerative medicine, development of organ on a chip platforms for chemotherapy induced cardiotoxicity. And she's also a Fulbright Fellow and has worked at Harvard, MIT Health Science and Technology departments towards the development of biomimetic cardiac tissues, cancer models, osteochondrocytes interface. Now, her contribution in the field of tissue engineering has been quite well recognized by peers and several other societies, and she's a recipient of the prestigious Vajpayee Shah Student Award at the SBAOI India, and she's also received a couple of awards for her presentation. And she would be, introducing a lot about her work to us today. So I hope everyone's quite excited, and even I'm quite excited to bring forward Dr. Shreya Malhotra here. And I'll just have her here. Hi, Doctor. Hi, Dr. Shreya. So we're very glad that we have you here, and I hope you're doing fine, and I hope you're as excited as we are, for this session. And the dais is yours. The moment you say I can present, you can present. I will allow your screen on the floor. Yeah.
I can present.
Yes. All right. You can go ahead.
Yeah. So thanks a lot, Aishwarya, for introducing me today to the audience. And I would also like to thank Altum Technologies for inviting me for this talk as a guest speaker today in their scheduled program. So, as much said that today I will be giving a brief overview of how we have tried to incorporate 3D bioprinting techniques in developing human cardiac tissues. And with this, I would like to now start, give a brief review of my work. So, So, as we know that in the recent years, 3D, with the advent of state-of-art manufacturing techniques and its applications in the field of tissue engineering, has enabled the 3D bioprinting of human tissues that have intricate architecture that is similar to the native human organs. Now, these kind of 3D tissues that are developed using 3D bioprinting methods have several advantages over the traditional fabrication methods, because the 3D bioprinting method allows for the development of biomimetic tissues that have spatiotemporal geometry that is similar to the native tissue. These kind of tissues using these techniques can produce, have high reproducibility, they have high feedability, and then we can develop multi-lineage tissues where different kinds of cells can be encapsulated within the different spatiotemporal geometry. And it also forms the basis of patient-specific graft fabrication. So using this 3D bioprinting technique, there are two main fields that have adopted this technique in the field of tissue engineering. First is the tissue regeneration, where these 3D bioprinted tissue constructs are considered as a replica of the native tissue and are used to fill the defects or the injured tissues, or replace the injured tissues in the human body, where they are expected to regenerate the lost tissue as well as to perform the function of the lost tissue. On the other hand, the other field that is looking forward to these 3D bioprinted organs or the human constructs is the organ on chip platform system, where these 3D bioprinted human tissues are considered again as a replica of the native tissue, and when it is coupled with a sensing platform or an on chip platform, it can form a basis of personalized medicine as well as drug discovery and drug screening processes. So, with this introduction that I have given here for 3D bioprinting, I would be taking both these fields, that is the tissue regeneration, where we will be talking about cardiac tissue regeneration specifically, as well as the organ on chip platforms, where we have tried to use these cardiac organoids or cardiac 3D bioprinted cardiac tissues for drug testing. So I will be moving forward with these two platforms in cardiac in my where I will showcase my work. So in the recent years, the need of engineered cardiac tissues has increased manifold, and this is mainly because of the first thing, that is the myocardial infarction, which is commonly known as heart attack. So, myocardial infarction specifically refers to an irreversible myocardial injury where a large amount of cardiomyocytes are basically your heart cells, which are lost in large numbers. Now, these cardiac myocytes, because they are terminally differentiated cells, they do not regenerate, and hence the heart will not be able to repair itself like how your skin repairs. So in that case, we obviously need a graft, or we need to introduce new types of cells into the heart tissue. So also there are several current modalities. What happens is that the current modalities that are present are the pharmacological approaches, that where we give drugs to the patient, that but they do not work on the remodeled heart tissue. So these kind of pharmacological approaches, they are mostly elevative and are not curative. Similarly is the case with angioplasty and stents, that they do not work on the remodeled heart tissue. So we have left ventricular assist devices, which actually do not work on the remodeled tissue, but they actually help in augmenting the function of the heart, where they help support the heart in its beating behavior. So in but they have their shortcomings in terms of the secondary infections and the thrombosis which is associated with the LVADs. And finally, we come to heart transplantation, where heart transplantation is obviously the gold standard treatment for myocardial infarction, but is quite limited by the paucity of organ donors. So, therefore, there is an urgent need of engineered tissues. The other sector that is looking forward to these engineered cardiac tissues is the pharma sectors, where futile years spent over collecting the clinical data, as well as your animal studies and the time consuming and expensive animal studies that are involved. To overcome all these issues, the pharma sector is looking at these 3D bioprinted organs, or tissue engineered organs, as a less time consuming and a less hassle free approach for drug discovery, as well as an approach that can form the basis of patient specific treatment. So with this, we do understand that there is an immense need of developing cardiac tissues, but in order to engineer the cardiac tissue, we need to understand that there are four different stringent requirements that need to be met for developing cardiac tissues. Now, in order to bioprint, or in order to fabricate human tissues, they require to be nature inspired. The meaning of nature inspired is that they need to derive their design or fabrication cues which are from the native tissue. So here the four stringent requirements that I just mentioned include: first is the anisotropic structure, where the heart muscles, they actually are in the rotated in the form of figure 8, which helps them to contract in two type of directions. So this kind of rotating structure, it provides heart quasi-lamellar or an anisotropic structure. So this kind of structure, mimicking this kind of anisotropic structure is really important. Second is the dense vasculature, where we know that the heart is profusely supplied with blood vessels, which and these blood vessels provide oxygen to the continuous beating of heart muscles. So that dense vasculature is also a prime requirement for fabricating or engineering cardiac tissues. And the third is the conductive Purkinje fibers, which help in the beating of the heart tissue. And lastly is the immunomodulation or the immunoacceptance, where these cardiac patches or these kind of cardiac tissues, if we are engineering and then we are looking forward towards a regenerative approach, these kind of patches should have the ability to modulate the hostile mi environment, because the mi environment or the heart, right after heart attack, the heart environment is quite hostile. There's lack of oxygen, there is lack of nutrients, there is infiltration of the immune cells, macrophages. So if we are going to implant a tissue there or a graft there, it has to have an immunomodulation property, so that it can secure itself by the time the host tissue is able to integrate with it, and it survives the implantation process. So with this the four stringent requirements, we try to use 3D, or we try to employ exploit 3D bioprinting technology for developing 3D cardiac constructs, which can be used for both regeneration and on-chip model construction. So here we have tried to formulate our own suitable ink that which could meet the stringent requirements of being biocompatible, biodegradable, should have properties, especially the mechanical and elastic properties similar to the native heart tissue, because it has to undergo continuous beating behavior. It should be clinically transplantable, and it should have low immunogenicity. So using based on this criteria, we designed the bioink, and then using this bioink, we have first tried to develop cardiac patches which can modulate the hostile environment, as well as augment the function of the beating heart tissue. And in the other approach, we have tried to again 3D print vascularized cardiac models that can be used along with on-chip platforms, and can form the basis of sensing systems, where they can be used alone or coupled with different organs. And then we can study the effect of drugs, and also study the drug-induced cardiotoxicity, or if there is a therapy that we are using, so, or a theranostic method, so we can study if there is any cardiotoxicity that is associated with these kind of drugs. So moving ahead, I will first talk about the regeneration process. So here we have tried to formulate a bioink comprising up of three polymers, which are silk, peg, and gelma. And this bioink follows a dual cross linking step to fabricate 3D constructs. So these bioink, we optimize this bioink to in a way so that we could actually print structures that had microfibrous structure, which was similar to the native heart ECM. And we also characterized the bioink, for its rheological properties, where we found that because the components of the bioink were such that they followed temperature based gelation. So we characterized to find the optimal printing temperatures, which was around 25 degrees Celsius, which is quite like the room temperature. And the bioink, we see from the amplitude sweep and the viscosity studies that it had a shear thinning nature, and it as well as had thixotrophic properties, where we talk about that when the ink is extruded from the needle, it has a different mechanical property. So as we can see here, before extrusion and after the extrusion from the needle, under pressure, there is no change in the viscosity as well as in the modulus properties of the bioink. So after extensively characterizing the bioink, we then use this bioink to fabricate these kind of 3D bioprinted patches, which were more uh, uh, tough and flexible as seen here. And we also characterize the mechanical properties of these cardiac patches, or the cardiac tissues that we developed, both in the static and dynamic system. And we found that these patches displayed mechanical properties or modulus values which were similar to the native heart tissue. So a heart tissue has around 30 to 45, 50 kPa of Young's elastic modulus. So, these kind of patches that we developed were similar to the native heart tissue and were better than the gelatin-based bioinks that have been previously used or are currently or mostly used for developing cardiac tissues. Also, in the dynamic system, we could see that up to 40 cycles or 50 cycles of continuous strain, there is no change in the mechanical properties. So these kind of cardiac patches developed using the bioink were really tough and could undergo several continuous compression or elastic tensile stretching. So they were quite tough. And then we also characterize these patches with for their usability for cardiac tissues reconstruction, where we seeded these kind of constructs, as you can see here. So what we did is, here we fed a CAD model. So we designed a set of G codes ourselves using MATLAB, and then we fed it into the bioprinter system, and we were able to design a construct which was similar to which had a structure which was quasi lamellar, which was similar to the native heart tissue. And then when we seeded this construct with human cardiomyocytes, and these cardiomyocytes are nothing but sorry, with rat cardiomyocytes, they are nothing but the cardiac muscle cells that we have got from the primary rat tissue. We seeded them. We saw that the 3D these cardiomyocytes had formed a 3D organ, as we can see here. It was densely populated, and each of these cardiomyocytes were attached along the microfibrous structure. So, saying that we could get a 3D construct, and we could define their arrangement as per the design and of the construct that we were giving through the bioprinter. So we also characterized for the functional properties of this 3D construct that we was formed on seeding these cardiomyocytes, and we found that these kind of constructs would displayed cardiac specific markers, which was important for the functioning of the cardiac tissue. And we also performed immunostaining, where we could see that these kind of cardiomyocytes, they had a good sarcomeric structures, and they were well connected using their connexin 43 gap junction proteins, and they had a beating rate of about 70 beats per minute by day 8. Similarly, for the proof of concept, we seeded them with human iPSC derived cardiomyocytes, and by day 7, we could have a beating rate of which was very close to the human tissue, that was 75 beats per minute. So, being successful with these kind of cardiac constructs using the bioink, we then tried to, move towards the more stringent requirements that were left over. That is, to talk about the vascularization part, to talk about the, conductivity part, and to talk about the immunomodulation part. So, as we know, I just want to give a brief overview that the MI environment is quite hostile. It is oxygen deficient. There are a lot of macrophages that are polarized, that are that are pro-inflammatory, and there is infiltration of the fibrotic cells, or the myofibroblast, which deposit a lot of ECM as a part of self remodeling of the heart, where the cardiomyocytes are lost. So in that case, these are the three main conditions that are seen in the hostile MI environment. And it was proposed that if we can modulate these three conditions by replenishing the cardiomyocytes using a 3D bioprinted or any tissue engineered graft, and by reducing the cardiac fibroblast at the heart tissue, or by polarizing the macrophages, we could develop an ideal cardiac graft. So here, using this technology, using 3D printing, we have tried to use a conductive bioink, which has nanofillers such as CNTs, and using these, we encapsulate UVEC biocells, UVEC cells. So UVEC cells are the endothelial cells that form a vascularized construct, and this vascularized construct is then kind of matured in a reactor to form the microvascular or the channel like structure in the native tissue, and it is then seeded with the cardiomyocytes. And these constructs are then added with IL-10 and calcium peroxide microspheres, which provide the oxygen releasing as well as the immunomodulatory properties. So we characterized, as I said, that the bioink consisted of carbon nanotubes. So we completely characterized this construct, and we could see that the carbon nanotubes which we included as a bioink acted as which was similar to the Purkinje fiber cell, which was similar to the at the fractural point of the conductive fibers in the native heart tissue. And the addition of these carbon nanotubes as a bioink component also enhanced the electrical as well as the mechanical properties of the construct, which could match the native heart tissue. So we also characterize these. So once we printed these UVX laden bioink to form a 3D construct. So this 3D construct which we had fabricated had a quasi lamellar structure which was similar to the native architectural of the heart tissue. So using we could easily get it, using a set of G codes, and using an extrusion printer where we could get these kind of fine microfibers as well as the arrangement in a 3D environment. So when we got these kind of vascularized cardiac constructs and we cultured them, so we found that by day 14 in a perfusion reactor, they had actually started to form a vascular or a vessel-like structure that could be seen by the different staining as well as gene expression studies, and they had a good survival as well. And then, once these kind of vascularized channels were developed inside, we seeded them with cardiomyocytes, and these cardiomyocytes were then seen to attach, along the fibrous structure of the patch, and they were viable. Further, they also displayed the properties that were similar, which were important for the maturation of the heart tissue in the forms of the sarcomeric structure that is formed, the gap junction proteins that are formed, as well as, when you talk about the gene expression, the all the uh, genes that are responsible for the functioning of the heart tissue or the cardiomyocytes, that is the actinin, cardiac troponin, which are the sarcomeric proteins, connexin 43, which is a gap junction protein for electrically coupling the cells so that they are beating at the same rate, and then alpha MHC, which is also sarcomeric protein. So all of them were displayed. There was an enhanced expression of these genes we cultured these kind of when we cultured cardiomyocytes on a ink that had a carbon nanotube filler. So also we could see that there have been reports that when we use carbon nanotube fillers as a component of bioink, there is an enhanced expression of this pathway, which we talk about, the mechanotransduction pathway, that is the YAPTAZ pathway. And we did see that there was an enhanced expression of integrin alpha 7 and RUA, which are the downstream signaling molecules. So we could say that if, in the bioink that we are using for cardiac, if we use a carbon nanotube based nanofiller, we could upregulate the mechanotransduction based behavior of the cardiomyocytes and also their maturation. So we also in we performed some patch clamp assay, and we found that when we seeded these constructs with cardiomyocytes, and externally we stimulated them in an electrical field, they could promote a ventricular like contraction, which into the cardiomyocytes. And this could happen this by having only a nano conductive filler, especially the CNTs. This was not possible in case of graphene or reduced graphene. So here when we stimulated them, as you can see in the video, we had a regular beating contraction. The beating rate was enhanced in the presence of simulation to around 130 beats per minute, and the beats were more regular by day 7. So we could get when we are using CNT as a part of a bioink, we are actually promoting a more rhythmic and a contracting, more synchronized contraction in the cardiac tissues that we are able to 3D print. So further, because we also added IL-10 and IL-10 based microspheres as well as calcium peroxide based microspheres, we could see that the 3D bioprinted cardiac tissues that we had developed were quite viable in the hypoxic environment. As you can see here, in the hypoxic environment, when we had the calcium peroxide microspheres, they had metabolic activity which was similar to those cultured in the normoxic environment. So which meant that whenever we will be using these kind of 3D bioprinted cardiac tissues, and we will implant them in a hostile environment where their already an MI has occurred in the heart, these kind of patches are at least going to survive by 5 days over there. The cells will maintain an activity which we would have seen similar in a in vitro condition, and this time would be enough for the host cell to take over the implant that we have taken. The implant might be able to integrate with the host tissue, and then the vascularization may overcome uh, uh, all the fibrotic tissue and all the hypoxic level that has been condition that has been formed at the MI region. Also, we also Then when we also characterize these kind of cardiac patches for their immunomodulatory behavior. So immunomodulatory, we mean that, when there is a environment where hypoxia has occurred, or the tissue necrosis has occurred, the cardiomyocytes are dead. So you have these kind of cells that infiltrate there, and they kind of disrupt the surface over there, and then they are they promote kind of inflammatory molecules, downstream signaling. So here, when we if we are trying to put a patch over there, that patch might not be able to survive because of the inflammatory molecules or the inflammatory cells that are there. So in that case, we can modulate those kind of cells to be anti-inflammatory, which is the M2 phenotype of the macrophages that we have tried to here. So using this kind of microspheres, we could actually modulate these kind of macrophages, and we could get immunomodulation properties. And then when we check these kind of bioinks, when we implanted them in a subcutaneous model in rats, we could see that definitely there was no immune response in the with for the bioink alone. With the bioink having carbon nanotubes as conductive fillers, as well as having these kind of macrophages. And further, we could see that especially in the constructs that were having calcium peroxide microspheres which were incorporated, there was initiation of angiogenesis, which meant that yes, the host tissue vascularization was actually infiltrating into the cardiac tissues. So we really feel that this kind of the thing cardiac patches, or which we have been able to develop, can actually help in regenerating human tissues, as well as animal tissues as of now, for regenerative purposes. So moving next, we would using this kind of suitable bioink, we are also trying to use the additive manufacturing method for developing on-chip models. So we all know that a large number of chemotherapeutic drugs are available in the market, and most of these chemotherapeutic drugs are associated with some kind of cardiotoxicity or ill effects or side effects on the heart tissue. So in a way that we can screen these drugs before they actually go into the clinical trials, we are trying to develop these kind of on-chip platforms. Further, the animal models that are currently being employed for these kind of drug testings are have different pharmacokinetics as compared to the human tissues. So these systems can provide humanoid organs which will be able to map the pharmacokinetics, which will be similar to the that of the human. So therefore, these kind of platforms are better in terms of mapping the cardiotoxicity that is related to the drugs that are coming out, as well as for the drugs that are available in the market. So in this respect, we have first tried to develop the vascularized cardiac tissue, and we used a different kind of technique, that is an embedded gel based technique. And in this type of technique, we took a supporting gel, which is which acts as a support or a bath gel, and that bath gel was consisting of cardiomyocytes. And using another bioink, which was gelma gelatin that we have used here, we used UVEC cells, and this bioink was used to embed vascular channels within that kind of bath gel, which was having cardiomyocytes. So we could actually get these kind of micro channels within a bath of cardiac cells. So we characterized the bath gel as well as the channel bioink over here, and we found that the bath gel had mechanical properties and porosity. That means that it could allow in the exchange of nutrients and could provide properties that were similar to heart tissue. And we also characterized the channel bioink, which was also shear thinning and could help in the embedding of channels around 100 micron, which was similar to the native heart tissue micro vasculature structure. So we also characterized them using live dead assays, and we found that the cells were viable. And when then we characterized them using functional assays, and we found that there were expressions of CD31 genes. They had formed the channel-like structures, as you can see here, HUVEC, and the cardiac tissue was also mature, as seen by the troponin T. And we could see here the formation of perfusible channel and a continuous and a rhythmic beating heart tissue. So as a proof of concept, we first try to before putting them along with any kind of chips, we first try to see if in a perfusion system how these kind of 3D models or 3D bioprinted cardiac tissues could react to drugs. And we found that when we used doxorubicin as a model drug, by the addition of drug, there was obviously a reduction in the beating rate, and there was an enhanced expression of troponin I, which is a marker for cardiac for drug induced cardiotoxicity. And there was a decrease in the expression of cardiac troponin T, which is a cardiac marker. And also, as by images, we can see that in the presence of dogs, the cells have actually disintegrated, and they have shown reduced beating behavior. So once we had you know cleared that in a perfusion system these kind of cells, on the action of drug, could be used as modeled cardiac tissue organs, we then tried to club them with a breast cancer tissue and see if we could study the chemotherapy based cardiotoxicity in these kind of on chips platforms. For this purpose we developed a dual chamber reactor where both kind of tissues, where you have a tissue from a healthy patient and you have a tissue from a fibrotic patient. So we tried to 3D bioprint these kind of tissues, and then these tissues were individually cultured with breast cancer cells and were used to study chemotherapy agent based cardiotoxicity. So briefly, we designed this kind of a sensing system where an immunosensor based, which was based on aptamer based sensing, was designed to and was then coupled with this kind of a dual reactor, and then by the action of drug, we could actually see, sense in our system, where if there is any toxicity. So we use the same embedded gel based system which I described before, and this kind of embedded gel had these cardiomyocytes. These cardiomyocytes were then treated with TGF beta 1 to form a fibrotic cardiac tissue. So we checked the effect of TGF beta 1 to see if how the fibrotic cardiac tissue was formed, and we could see that by gene expression and immunostaining that there was enhanced expression of the fibrotic markers, that is collagen, alpha SMA, and vimentin D. So therefore, which meant that we could form both healthy and cardiac tissues using the embedded gel bioprinting method and then treating them with TGF beta. We then formed the breast cancer tissues, where we formed these kind of tissues in the microvels, and then these PDMS spheroid-like structures were then put inside a gel to form a organoid kind of system, which displayed good viability as well as an expression of HER2 marker, which is a breast cancer marker. We then cultured both these healthy cardiac tissues with the breast cancer tissues and fibrotic cardiac tissues with breast cancer tissues, and then we cultured them in these kind of dual reactors where we stimulated them for best oxygen concentration, flow rate, and drug concentration, and then we coupled them with these kind of an immunosensors. So these immunosensors were formed actually out of gold, because of its enhanced stability, and they were functionalized with aptamers using an ADC NHS based cross linking method. So these aptamers were then these sensors that we fabricated were characterized, and we found they were highly selective, they were highly specific, and they could measure around 0.08 picogram per liter of biomarkers, which is way below the presently used sensing methods, that is the ELISA based sensing kits, that is one which can give around 1 nanogram per ml. And when we clubbed these kind of fibrotic tissues with the sensor system, and we used our model drug doxorubicin, and we compared these our readout values with the ELISA kits, we could see that we could actually get a difference in the biomarker concentration, which means that on the action of drug, we could actually understand, if there is cardiotoxicity both in a patient that is having a disease condition of the heart, or in a tissue that is derived from the patient that is quite healthy. So we predicted these outcomes, where from these studies, and we found, as I said, that using our sensing system and on-chip platforms that we developed using a 3D bioprinted vascularized cardiac tissue, as well as the aptamers that we had, the sensing multiplex aptamer-based sensing system that we had developed, when we are coupling them together, we could actually develop a system where we could study, or we could catch a difference in the biomarker concentration in case of fibrotic patients, which is not easy to catch in case of the presently used sensing systems. So this sensing systems we propose that may form the basis of low cost and high precision platforms for patients or personalized medicine kind of systems. And coming to conclusions, so here we have used, as I said, that we used non-mulberry silk. So silk, here we have tried to promote silk as a optimal biomaterial ink for fabricating cardiac tissues, and using this kind of a bio ink, we could develop both vascularized as well as avascularized cardiac tissues, and these vascularized cardiac tissues could help in regeneration. We could impart specific properties like conductivity in case of by using cardiac nanofillers, that is CNT nanofillers. We could enhance the immunomodulatory properties by adding IL-10 microspheres, as well as oxygen releasing property by using calcium peroxide microspheres. So using these kind of properties which could actually enhance the performance of these 3D bioprinted cardiac grafts, so such that they can modulate the host microenvironment for enhanced graft acceptance. And finally, the 3D bioprinted organs that we have developed, or the 3D bioprinted constructs that we have developed, they could be coupled with a multiplex after sensing platform and could form the basis of personalized medicine. So some of the we have been able to patent our bioink. We have filed a patent for our bioink that we have used here, and our work has been highlighted, on several journal covers as well as on in media. And finally, coming to acknowledgments, I would like to acknowledge my mentor, uh, my Fulbright supervisor in Harvard and MIT, who has helped me extensively, and obviously my mentor now at IIT Kanpur, Professor Ashok Kumar, for giving me this opportunity. I would like to thank all the collaborators, all the people who have been a part of my team, and finally the funding agencies, without whose generous funding this extensive work would not have been possible. Thank you. So I'm open to any questions that are there regarding my presentation.
Um, Thank you so much, Dr. Shreya. I think this was a very wonderful and informative session. I will slowly come down to adding few of the questions that have been asked by people.
Yes.
Just a minute, I'll just scroll through. Okay, so here there's a question saying, what are the challenges faced while bioprinting? So could you kindly elaborate on the general challenges that we face during this process?
So the general challenges that we face specifically with here, I've used extrusion-based bioprinting, is that you have to get the microfibers. So you should have the correct viscosity of the bioink so that you can get that microfiber structure. So the bioink composition is one of the major thing that needs optimization and is also a challenge. And once you are set, you are able to get a bioprinter, you are able to get a microfiber structure. Then the next thing that comes is how you adjust the printing temperature, you adjust the printing speed, flow rate. So those parameters that need to be adjusted and optimized for the bioink. Apart from that, in extrusion, what we try to do is that what happens is that for most of the bioinks, the if we use highly polymers that can be cross-linked into a tough structure, we lose on cellular viability. But if we use a bioink composition which can form a soft structure where there is a lot of interpenetrating space, and the cells are able to move around and form are able to interact with each other. At that time we lose the we lose the structural ability of the bioprinted construct. I mean, you cannot see well defined boundaries of each layer that is formed. So that is one challenge that we have to face, and that is optimized between the bioink and the bioprinting process.
Okay, so there is this question about how 3D bioprinted printing is used in nanotechnology of a tissue.
I'm sorry, but how, I mean, I didn't understand this question. So is
Okay, I am not sure if they'll be able to elaborate on this. Okay, maybe we'll skip this for now. I'll check for others. Okay, this could be a slightly off your topic question, or maybe could be helpful here. But yeah, can we test various concentrations of drugs in parallel to check the efficacy?
Yes, I mean we can do that. I mean we usually here I have just tried to develop one dual chamber, and we have just connected it through a one pumping system. But we can have several chips because I showed you that the sensing platform has around 20 electrodes. So it is a multiplex sensing platform. So we can have multiple of these chambers, and then we can check different concentrations of drug with the same sensing platform. Yes, we can
All
do that.
right. Okay. Um, Another being, what are the advantages of using these kinds of bioinks in compared to other bioinks? So I think they mean distinctly about the material you have used versus general set of materials.
So here, what we have tried to do is that we have tried to keep in mind the specific stringent requirements of a tissue. So it's not about the bioink. You have to match the bioink properties with the tissue that you are looking for. It may be a regenerative purpose, or it may be for an organ on chip platform. But every tissue, for example, if you're talking about a heart tissue, it needs to have certain kind of elastic properties. It needs to be, it should be not too tough, and it should not be too soft, because you have to allow the cells to contract. So these kind of parameters actually define the bioink composition that you are using, that or we might be using. On the other hand, when we talk about bone, we can have the liberty to move into a more thermoplastic type of polymers. We can use polycaprolactone, we can use different synthetic polymers as well. So it depends more on the kind of tissue that you are looking, and then you match the properties with the bioink.
All right. Yeah. Thank you. I will go to the next question. What material properties were kept in mind while selecting the carbon fiber nanotube?
So here, basically, carbon nanotubes that we have used here, carbon nanotubes, basically we tried to we use them to enhance the conductive properties so that we could get a better beating output from the tissue that we had developed. Moreover, carbon nanotubes enhance the mechanical properties also. So, and when we saw through SEM, we could see that carbon nanotubes were actually mimicking the carbon or the Purkinje fiber structure at the fracture points. So it's the kind of as I spoke earlier, that that kind of conductivity, when you are stimulating the cardiac tissue from outside, it is required. It is a stringent requirement of a development of a cardiac tissue. So keeping that in mind, we used carbon nanotubes as a bioink filler or a bioink material. And also, when, as I said, that the beneficial factor is that when we stimulate these kind of carbon nanofibers base substances or 3D bioprinted constructs using an external magnetic field, and then when we have cardiomyocytes seated on these constructs, we could see that they could enhance the this ventricular phenotypic beating behavior. So that also we found that they could enhance the cardiomyocyte maturation. So all these parameters have been beneficial, when we use CNTs.
Okay. I will go down to the next question. We can always stop whenever you think, you've answered
Yeah.
too many questions. So, how was the cardiac tissue stimulated exactly? Basically, the mode of stimulation.
So we have tried to we generally use an AC voltage supply to stimulate them, and we use 1 hertz and a square wave pulse based stimulation method, and then we use the electrodes that we where we are culturing them in an in vitro system. So we use those kind of plates. They are being customized to have those kind of electrodes, and we use carbon or platinum based electrodes to
All
stimulate them.
right. All right. So I'll take this question, and after that, you can decide if you want more questions.
Yeah.
So yeah, how is it quantitatively biomarker expression was analyzed in cardiac tissue engineering using this technology? Can should I repeat the question?
Yeah, I understood.
Yeah.
So basically, what we are doing here is that I think, we developed this multiplex sensing platform. This multiplex sensing platform has a couple of sensors that are metallic sensors, and we have used an impedance based method. sensing method. So basically, whenever a biomolecule will attach on the surface of an electrode against the aptamer that we have applied, any biomarker that will attach, there will be the change in the impedance value or the resistance
Mm.
impedance value. That is, the electric current versus voltage graph will show some changes. We will get changes in the Nyquist curve. So depending on the change we get in the graph, we quantitatively, using the standard graphs, we compare it and then we get the quantitative biomarker expression.
All right. Thank you. Um,
So I can take a couple of more questions and then we can.
yes. One second. Okay. So, how were the microchannels evaluated or tested?
The microchannels in terms of in which micro, are you talking about the UVEC channels? Are we talking about the bioprinted channels?
I'm not sure, but maybe you can elaborate about both since we have, yeah, since
So
we
with
have to.
Yeah, so where we if we talk about the perfume, the channels that we printed using our embedded ink, we used a couple of methods. We used SEM, we used microscope based technology also, and then we have also done these kind of I would say staining based methods, where we have taken multiple again microscopic images and compared them, and then also SEM images, and then cross check them together.
Okay, so you've used the imaging methods in general. Okay. One second. Yeah. So, what is the survival rate of the cardiac patches?
So as of now, when we use seed these patches with rat cardiomyocytes, because they are primary cells, you cannot have them for long. We are able to get a beating patch for around 10-15 days, but standalone in vitro. But when we have tried to use human iPSC derived cardiomyocytes, that is stem cell based, human
Hmm.
cells, human cardiac cells, we differentiate them, so that we are able to actually have them beating around 20-25 days in culture. And but that
Okay.
is in culture, and then they are standing alone, but they will have a better survival rate once they are implanted, because they will be able to integrate with the host tissue, so their survival rate will automatically enhance.
An additional question to that from my end. So, how long did it take for the regeneration or the integration as such? I might have missed it, but I would like you to elaborate a little more on that.
Yeah, so with our materials that specifically we had where we've used these kind of patches in, we use subcutaneous tissue implantation method. We could see that by day 14, there was infiltration of cells in major areas of the patch, and I think by 21 days there will definitely be a good integration with the host tissue.
All right. So the another question from my end again. So, how exactly do you see this going forward? I mean, like, of what I understand, this would have gone to the animal testing, preclinical testing. How much do you think would it require in terms of progression towards getting human, at least end user sort of a product? So what do you think are the ways we can get there? What are the challenges you think we would have when it comes to going to that point?
So there are, right now, we are moving forward with animal studies. We are actually developing MI models in animals, small animals, and we are trying to implant these patches there. So, there are some, challenges that have to be met. How are we going to surgically implant them? What kind of bioglues that we are using along with the 3D bioprinted constructs, and if how is it going to repair the heart wall? Is it actually just going to provide just helping in that area and not actually helping in the function? So those kind of parameters we still have to do. We have just done a very preliminary study, and we have found, at least in the in vitro conditions, we have been quite successful. And these kind of grafts are non-immunogenic, but how they actually function at an MI region is one of the studies that we have now taken forward. So yeah, I mean, there
All
are still
right.
challenges.
All right. So maybe we'll take two more questions if that's all right.
Yeah, okay.
Yes, so this is, I think, a generic question. It could be slightly repetitive, but I will still read it out for you. How can we be sure that these carbon tube and cardiac cells be successful in real human model, as we know the complexity in the immune responses?
So, I mean, there are a couple of studies where people have used these carbon nanotubes as drug delivery platforms already, to deliver to tumor cells or uh, these kind of you know so I don't think using carbon nanotubes, I mean, of course, there is not an FDA approval yet for carbon nanotubes, but there are both there are a lot of people that agree that carbon nanotubes can be used, and there are also groups that agree that they also say that carbon nanotubes can have some problems, where they are so fine that they can actually rupture the cells. So that is, we are far away, because using carbon nanotubes is not yet FDA approved. So this is kind of something that is, we are quite ahead of the present time in kind of our research. And secondly, using cardiac cells, yeah, I mean, of course, because here we have tried, in case of humans, we are using iPSC derived cardiomyocytes, and there are clinical and ethical issues, concerns that are there with using iPSCs. So yeah, as of now, I mean, we are quite ahead of the time that the
All right.
regulations are there.
Okay, so I'll take out this one question. I think it's been repetitively asked. So it says, please explain about bioprinting usages in cryogenics.
Uh, so there are different kind of bio inks that can be used for cryo bioprinting methods. I mean, there recently, I've gone through a couple of methods, where they are actually using these bio inks. You print them at cryo temperatures, and they're quite stable, and you can get a 3D structure. But definitely the bio ink that I have proposed here cannot be used at a cryo uh, conditions. So
All right. Yeah, I think we're almost towards the end of the questions. All right. So, thank you so much, Dr. Shreya Malhotra, for joining us. Thank you
thank
for
you,
taking out
Ashwini.
so much
Yeah.
time. Yes, thank you for taking out so much time, and we really enjoyed the session. I especially loved the examples that you've given in terms of tissue regeneration or organ on chip model, and I hope we can get in touch further and learn and understand more about this. So, thank you so much, Dr. Shreya. Um, yeah, so I will take you off the session for now.
Yeah.
Yeah, thank you so much. Yeah,
Thank
bye.
you. Yeah. Bye.
Thanks a lot, everyone, for joining. And please remember, we are available on altum.com. Kindly follow us on LinkedIn, YouTube. Please share this link to anybody possible. And we hope to continue giving you such detailed explanations on tissue engineering as well as other platforms. Until then, please. Stay and join us on our next session, which is tomorrow at 7 p.m. again, which is on the examples of bioprinting in general, the cases as well as the biomaterials developed and used entirely in the process. So thank you so much. Have a very nice day ahead.